Okay, right now what I'm going to do is I'm going to present a video lecture about Section 8.2. At this point in time, we are hoping that you have read through Section 8.2 and you've gone through the outline that was provided to you by your teacher. This video lecture is basically for anyone who still needs a little extra explanation about what the section is about. You'll notice on the class website, right above this video link, there was a link that said 8.2 notes. If you wanted to print those out and have an extra set of notes available to you, by all means, please do so. Those notes would be a good idea if you were going to follow down and write what I was saying down, because those notes follow this PowerPoint exactly. What Section 8.2 is all about is just DNA. Simple, how did we learn what the structure of DNA was? What do we use DNA for? Now, for DNA, we know that it is used as a genetic material that makes all of us who we are. Okay? What you may or may not have known is that DNA structure is the same for all organisms. So our DNA structure is the same as a mouse's DNA structure, as is a fruit fly. We all have the same type of structure. But where did that structure come from, and what does that structure look like? Well, to understand that, we need to understand what is DNA. DNA is basically a sequence of nucleotides put together. Think back to monomer versus polymer. Monomer means one, polymer means many. So a DNA would be a polymer. It is many nucleotides put together. So what makes up a nucleotide? A nucleotide has three different parts. It has a phosphate group, a sugar, and a base. In the case of DNA, we have a phosphate group on the outside, we have deoxyribose, our sugar group, and then we have one of four different nitrogen-containing bases. You'll notice at your picture that the phosphate group is a circular shape. It's connected then to the stop sign, or your sugar, and then it's connected to one of, again, four bases. In all of the examples that we will go through today, we will be referring to the sugar and the phosphate as the backbone, and we will be referring to the bases as the inside. Now, another way to think of backbone as well would just be outside. Okay, so let's take a closer look at what makes up this inside. Let's talk about the bases. Your DNA has four different bases. You're going to notice you have thymine, cytosine, adenine, and guanine. For simplistic sake and spelling purposes, we just call them A, T, C, and G. Those are our bases. Now, what we will go through with A, T, C, and G is you'll notice they're put into two different categories. You have your pyrimidine category and you have your purine category. This is important to understand because when we talk about how they pair, they match up. So a pyrimidine pairs with a purine, and a purine pairs with a pyrimidine. So how did we figure all of that out? Well, the first two people to put together a structure of DNA would be Watson and Crick. Watson and Crick came up with and designed a three-dimensional structure of what they thought DNA looked like. What they realized is that DNA is actually a double helix. And a double helix, to me, it's kind of like a spiral staircase. So if you saw this great big winding staircase, that would be the same idea as DNA. And what they discovered is that the sugar and phosphate, that backbone, that's this part. So the sugar and the phosphate are these outsides. Okay, so they make up that outside part. And then on the inside, they discovered it was the bases. So the A to T, that's C to G, that's all the insides. That's important to know because when we talk about how DNA is bonded together, we want the outside, that outside component to be super strong, but we want the inside to be a little bit weaker. And that's what kind of Watson and Crick figured out. Well, how did Watson and Crick figure out that it looked like this spiral staircase? Literally, it looks like as if you took a ladder and then you just started twisting it up. Well, let's look at the research that they used, because Watson and Crick did not do this all on their own. They used the work of many different scientists, and two scientists specifically was Rosalind Franklin and Erwin Chargaff. These were another set of scientists basically trying to do the same thing. They were trying to discover the structure of DNA. They just wanted to do it first. So Rosalind Franklin is an X-ray chromatographer. Basically, that means she took pictures of chromosomes using radiation. This ended up taking her life because she ended up dying of radiation poisoning. But before Rosalind Franklin passed away, she took a very important picture. It's the picture that you see in front of you. It's that X. And that X was the missing piece to Watson and Crick's puzzle. When they saw that kind of circular middle followed with all those dots stemming off from it, they realized that DNA wasn't just a straight line. It wasn't this way, that way, or there. It was this. It was that spiral. And that's what led them to put everything together. Now, once Erwin Chargaff saw Watson and Crick's structure, he came up and said, well, look at how the bases pair together. A pairs with T, and C pairs with G. So those are actually known as the base pairing rules. So if you look at the base pairing rules, no matter what, A will always pair with T, and C will always pair with G. Why is that important? Well, that's important because remember I said the inside of your DNA is weak? We want it to be able to break apart. We want it to be able to break apart because in Section 3, we'll talk about DNA replication. How do you make new cells? Well, your DNA has to copy itself. In order for DNA to copy itself, it has to break apart. So your DNA, which is this nice wound up structure, actually starts to split during DNA replication. You've got A's and T's, they always pair together. C and G will always pair together. So now, what happens for the next step is they kind of pair off again. And like I said, we'll talk about that more in Section 3. But the one thing I really want you guys to understand is that the main key, like if you could just memorize this one thing, memorize that A pairs with T and C pairs with G, and this is for DNA. And it's the same flip-flop. T will pair with A and G pairs with C. If I was to give you a sequence of letters, and that sequence of letters just included A, T, C, and G, could you match them up? And I know that each and every one of you could, because you follow that same coding technique. A pairs with T, T pairs with A. C goes to G, G goes with C, T goes with A, T goes with A, and A goes with T. So it all matches together. And that goes back to that single double ring structure. A single ring always pairs with a double ring, and a double ring pairs with a single ring. You won't find two single rings, and you won't find two double rings pairing together. All right, so let's kind of look at one of the last steps. It's what does it look like when the spiral staircase is unwound? When your DNA is unwound, you'll notice you've got your sugar phosphate, those are those blue, on the outside, and then you've got all your colorful bases on the inside. These are held together with covalent bonds. Think of covalent bonds, and if you don't remember them, they're your strong bonds. I've referred to it as your backbone because think of your skeleton system. Would you want your backbone to turn to mush? No. You'd crumble. You'd fall right down. So that's the same thing with your DNA. You do not want that backbone, those sides, to crumble apart. But because we want to be able to copy our DNA, we do want the insides to come apart. So they're held together, and you'll notice that's kind of my dotted line here. They're held together by a hydrogen bond. Hydrogen bonds, if you don't remember, they're weak. And they're weak for a reason. They're weak so our DNA can copy itself. Go back to the cell cycle. G1S, what happens in synthesis? DNA is copied. DNA could not be copied if that was a strong bond. We need it to come apart. At this time, that concludes section 8.2. If you have any questions over the structure of DNA, which was discovered by Watson and Crick, and or DNA-based pairing rules, A to T, C to G, please feel free to talk to any one of the biology teachers.